Good afternoon, everyone. My name is Lee Lind, and it's my pleasure to welcome you to the Thayer School Jones Seminar Series, and also to introduce my friend and colleague, Karl Griswold, who is our speaker today. As most of you know, Karl is an associate professor at the Thayer School. He's been with us since 2007. He got his undergraduate degree in chemistry in 1995 at Texas State University. Stayed in Texas, worked in George Giorgio's lab at UT Austin for both a doctorate and postdoc in chemistry, but then decided it was really too warm down there, and he needed to move north, and we were glad to receive him. While Karl's been with us, he's won an award in translational biomedical engineering from the Coulter Foundation. The New Hampshire Innovation Research Council gave him the Grand State Technology Innovation Award, and he's won a Teacher of the Year Award from Thayer School. He's co-authored more than 60 peer-reviewed papers and is inventor on 6 issued US patents, 1 foreign patent, and a lot of patent applications in the pipeline. He's been active as an entrepreneur, and in fact, took a couple of years to focus primarily on that rather than his academic activities. And Karl, we're really excited to hear what you have to say, and the floor is yours.
So Lee, thank you very much for the kind introduction. Let me see if you all are seeing what I intend you to be seeing. Great. So yeah, thank you for the introduction, Lee. And I have to admit that it is a little bit bittersweet. I'm disappointed that I'm not standing in the seminar room, looking each of you in the eye in person, but nonetheless, it's a real pleasure to be here with you this afternoon and to have the chance to tell you about some of the work that we've done on a collaborative project, a wildly successful collaborative project over the past many years. This is a partnership between Chris Bailey Kellogg's group in the Dartmouth Computer Science Department and my group here at Thayer, and then more recently, collaborating with some startup companies that we have formed. So pursuant to that point, before I get into the talk, let me make the following disclosure that I am a co-founder and current CEO of a group of affiliated biotech companies that are focused on design and development of next generation biotherapeutics, and conflicts of interest associated with my work at these companies is under management at Dartmouth, including making this disclosure. So work in my academic group as well as at the companies is really motivated by this just powerful reservoir of potential therapeutics that is embodied in proteins. So here in this slide, I'm just showing you a handful of the examples of therapeutic proteins that have contributed to a revolution in modern medicine. So this is an antibody, Humira. It's the #1 selling drug in the world. Over the past couple of years, it's sold about $20 billion a year by itself. Asparaginase is an essential medicine annotated by the World Health Organization to treat acute lymphoblastic leukemia in children. Alpha-galactosidase is a life-saving medication for children with congenital diseases associated with polysaccharide storage in lysosomal vesicles. Avonex has been a foundational therapy for multiple sclerosis for many years. Lumoxiti is actually a new innovative immunotoxin that's been engineered to treat leukemia in adult patients. And then today I'm going to tell you about a molecule called lysostafin that represents a potential next generation antibiotic. So I want to emphasize that proteins are nanoscale machines that just have unrivaled capacity to interface with biological systems and to affect beneficial outcomes in the context of addressing disease states and disease pathologies. Now, while they are really driving a revolution in modern medicine, as I said, they don't come without their own limitations and risks. And in fact, one of those limitations is that the biological origins of proteins renders them subject to surveillance by the human immune system. So here for these same drugs that I showed you over on the left-hand side, I'm now showing you the percentage of patients that develop anti-drug immune responses against these agents. So where do these immune responses come from? What's their source? And the origins of this it's really grounded in the fact that the human immune system is one of the most sophisticated and efficient molecular pattern recognition systems on the planet. And what I'm showing you here, rendered on the polypeptide backbone of these biotherapeutic agents, is shown in red are some of the patterns or high-risk patterns that might be flagged by the human immune system. And I'm going to kind of get into just a little bit of background on how the immune system recognizes these patterns and what it does upon recognizing those patterns. But maybe just to set the stage for this, I thought what I would do would be come up with maybe a more relatable analogy with respect to danger and pattern recognition. So I'm going to give you all just a second to digest this slide. There's a pattern recognition quiz for you. And the question is, which of these two snakes, number 1 or number 2, would you choose to cuddle with? The answer is A, snake 1, B, snake 2, C, both, or D, neither. So Megan, if you wouldn't mind, could you go ahead and launch a poll? If you all are interested in participating in an interactive poll as part of this Jones seminar, I invite you to provide an answer. I'm not going to give you very long because I'm strapped for time here, as I always am when I'm talking about my science. So Megan, why don't you go ahead and close the poll and maybe share the results? Snake 1, neither. Wow, okay. So for what it's worth, those of you who answered D, neither, you're of like mind with me. Those of you who answered C, both, you're answering along the lines of what my wife would say. She's a passionate wildlife biologist. Those of you who answered snake number 2, maybe not by the poll, but in your heads, I'm going to encourage you not to cuddle with snakes in the south of the United States. And those who answered snake number 1, right, I suspect that you're familiar with the mnemonic red. Sorry, give me 1 second here. Let me get the laser pointer. So red and yellow kill a fellow, whereas red and black venom lack, right? So this is a mnemonic to help people distinguish between a coral snake, which is a venomous snake that produces one of the more toxic venoms among snakes, and a scarlet king snake, which actually, right, is fundamentally, it's a snake. In fact, it has the same colors as a coral snake. It even has a banding pattern like the coral snake, but this distinction in the patterns between the two, so the proximity of the red and yellow in the coral snake and the red and black in the scarlet king snake, allow you to distinguish the fact that this coral snake is very dangerous. You don't want to be handling the snake, whereas the scarlet king snake might give you a bite, but it's not going to be life-threatening. So it turns out that the human immune system does this type of pattern recognition on a constant basis every day in order to protect our bodies from foreign invaders. But it does this type of pattern recognition in the context of kind of scoping out molecular threats instead of snakes. All right, so maybe just to put a finer point on this, I thought I would start with taking a look at all of our favorite virus right now, so the SARS-CoV-2 virus, and how we are all going to be rescued from this pandemic eventually, hopefully sooner rather than later, is that we're going to get a vaccination. We will be immunized with the spike protein from this SARS-CoV-2 virus. And so the idea here is that this spike protein is going to be injected into our bodies. And I'll note that actually there's some very cool technologies that are now being used for vaccination. So the exact form in which we are going to get this protein may differ depending on what vaccine you get. But ultimately, at the end of the day, we're going to get this SARS-CoV-2 spike protein injected into our bodies. And once that spike protein is in our bodies, it's going to be surveilled by different groups of immune cells that are in our immune cell repertoire. So there are a couple of different flavors of professional antigen presenting cells that are very relevant to this. On the one hand, we have dendritic cells, which can internalize that spike protein in a receptor independent, kind of non-receptor dependent fashion. they will unfold that polypeptide chain. So keep in mind that what are proteins? Proteins are just linear polymers of amino acids, so long linear polymers that fold up into very precise three-dimensional structures that have all of these wonderful properties from a drug development perspective. So the viral spike protein here will be unfolded by the dendritic cells upon internalization. It'll be proteolytically processed or chopped up into very small peptide fragments, so just pieces of the original protein. And those small peptide fragments or pieces will be displayed on the cell surface in complex with proteins called class II MHC. So those proteins are really important in the context of this talk today. Now, another group of professional antigen presenting cells are what are called B cells. B cells are ultimately the cells that produce the antibodies that are going to protect us. So B cells will also internalize this spike protein, but they'll do so in a receptor-dependent manner using their B cell receptors. Those B cell receptors are essentially the forerunners of the antibodies that will ultimately provide protection to us. But nonetheless, those B cells, again, internalize that spike protein. They unfold it much like the dendritic cells do, proteolytically process it, and display those exact same peptides on their surface in complex with class II MHC. Now, if indeed this peptide fragment represents a danger signal or a dangerous pattern to the human immune system, then a subject's immune cells, specifically their CD4 plus helper T cells, will have cognate receptors that can recognize that peptide displayed on the surface. And it's this formation of a ternary complex between a peptide fragment of the viral spike protein, class II MHC on the antigen presenting cell, and the T cell receptor on a CD4 plus helper T cell. That ternary complex formation drives a signaling cascade that results in T cell activation. These activated T cells can go on and interface with antigens with this dangerous pattern or dangerous signal peptide displayed on the surface of immature B cells. That will actually drive formation of plasma cells from these B cells. So the plasma cells are the form of B cells that just crank out large quantities of these high affinity antibodies that will specifically bind to the intact viral spike protein. And it's these antibodies that will bind to that spike protein on the surface of the intact virus that offer us protection. All right, so all of this is exactly what we are waiting for. This is what the immune system has been engineered to do, is to intercept threats and protect us from these external threats. So in the context of a viral infection, this is exactly what we want to have happen. But now let me just have a slight shift in perspective here. And let's imagine that we're not being injected with a viral spike protein vaccine, but rather you're a cancer patient, or you're a patient that has a life-threatening bacterial infection, and you need to be dosed with a protein drug in order to treat that disease. Well, it turns out that when you're dosed with that protein drug, the immune system can't distinguish between a drug that your physician gives you and the viral spike protein that you may get in a vaccination or that you may get exposed to by virtue of a viral infection. And so the exact same cellular and molecular recognition pathways that will provide protection to that SARS-CoV-2 virus once we're vaccinated can also drive formation of antibodies against a protein therapeutic that we're taking intentionally in order to treat a disease. And the formation of these anti-drug antibodies in the context of a protein therapeutic can cause all kinds of clinical complications, ranging from drug inhibition to altered pharmacokinetics, deposition of toxic immune complexes in tissues and vasculature, various allergic type reactions, including the possibility of life-threatening anaphylaxis. And from a patient's perspective, you may have to get pulled off of a biotherapy that was life-changing for you. And from a drug developer's perspective, it may be failure in phase 3 clinical trials after a company has already invested $1 billion, $2 billion, $3 billion in development of a therapy. All right, so the development of antibodies is great in the context of protecting us from a virus, and it can be devastating in the context of neutralizing a protein therapeutic. All right, so the interim conclusion here is that anti-drug antibody responses can undermine a biotherapeutic's clinical utility. And I want to emphasize that while it's the antibodies that manifest the clinical complications, in fact, it is the T cell epitopes that fundamentally drive formation of those high affinity anti-drug antibodies. All right, so what I want to do now is I'm going to shift gears and tell you about a second vignette that may on the surface appear unrelated, but you'll see that I'll make the connection. So one of the problems that we've been trying to address in my research lab for many, many years is the unmet medical need represented or embodied by the antimicrobial drug resistance crisis. And just to kind of put a point on part of what has gotten us to where we are with drug-resistant bacterial pathogens, I'm showing you here a timeline that was printed in a CDC report on antibiotic resistance. So on the top, I'm showing you the date at which various antibacterial chemotherapies for Staphylococcus aureus were introduced. And on the bottom, I'm showing you the dates at which resistance to these drugs was detected. So it turns out that drug-resistant staph predated clinical use, penicillin-resistant staph predated clinical use of penicillin. Methicillin was a next-gen beta-lactam antibiotic developed in 1960, resistance to that in 1962. Turns out vancomycin had a pretty good shelf life and it's still used, right? Developed in 1972, resistance showing up late in 2002. But these more recent therapies, and in fact, standard of care therapies now for drug-resistant Staph aureus infections, including linezolid, introduced in 2000, resistance in 2001, daptomycin, introduced in 2003, resistance in 2005, and ceftaroline, introduced in 2010, resistance in 2007. And so what this represents is there is an arms race between bacterial pathogens and the chemists that are developing antibacterial chemotherapies. And the fact that bacteria are so fast at developing resistance to the antibiotics that we introduce into the clinic has really painted us into a corner with respect to dealing with these infections. So here I'm showing you two graphics. On top, on the y-axis, is the incidence or the frequency with which drug-resistant bacteria have been isolated in the clinic from the years early 1980 to mid to late 2000s. And I'm showing you the trace for 3 different common bacterial pathogens. And the take-home message here is that over this time frame, there's been a pretty steady increase in the frequency with which these bacteria are found in drug-resistant forms in the clinic. Now, over that same time frame, the early 1980s to the mid to late 2000s, you'll see on the y-axis here in this bar graph, there's been a relatively steady decline in the number of new antibiotics being approved by the FDA. So again, this has really painted the medical community in the world into a dark corner with respect to dealing with these types of drug-resistant infections. Over the past 4 to 5 years, or maybe 4 to 5 years ago, there was a report that was commissioned by the British government and the Wellcome Trust that concluded that if no action was taken on this crisis, that by 2050, there would be 10 million deaths a year globally from drug-resistant bacteria. That is going to eclipse the rate of cancer deaths at that time. And from a financial perspective, if no action is taken, there will be an annual $1.2 trillion in excess healthcare costs. And by that time, there will be a cumulative $100 trillion loss in global GDP. Right? So this is a serious health crisis. It's something that is demanding attention. But again, partly because of regulatory issues, partly because of technical issues, and also financial issues, you see drug developers kind of abandoning the search for new treatments for these drug-resistant infections. All right, so we've looked at lots of different drug-resistant bacteria in my lab. One of our favorite ones to focus on is methicillin-resistant Staphylococcus aureus. This is a multi-drug-resistant Staph aureus bacteria. It's actually responsible for about 30% of all deaths in the United States from drug-resistant bacteria, and it contributes to about $2 billion a year in excess healthcare costs. So drug-resistant bacteria are bad. Methicillin-resistant Staph aureus, or MRSA, causes a particularly outsized impact on health outcomes and the healthcare system. So there's a desperate need to develop new therapies for these types of drug-resistant bacteria. And as opposed to further elaboration of small molecule chemotherapies, my group and numerous other groups around the globe have become intrigued by the possibility of leveraging biotechnology to address this healthcare crisis. And we in particular have focused on the possibility of using enzymes or nature's catalysts as novel therapeutics to treat these types of Staph aureus infections. And in particular, we focused a lot of attention on a molecule called lysostatin. So this is a 2-domain enzyme. It has an N-terminal catalytic domain that I'm showing here in blue, and this is effectively a pair of molecular scissors. And then it has a C-terminal cell wall binding domain that I'm showing in red. So this essentially turns the molecule into a guided missile that homes in on the bacterial cell surface. Now, what I'm showing below is the amino acid sequence, or the primary structure for lysostatin. Each one of the 20 natural amino acids has this 1-letter code for it. And I bring this up in part to emphasize that what I'm going to tell you about today is our efforts to engineer lysostatin to make it a better biotherapeutic. And how do we do that? We use genetic engineering tools to go in and change the amino acid composition of this enzyme. We swap out natural amino acids for our own choices of amino acids. And by doing that, we seek to affect subtle but important changes in the protein structure that will impart hopefully dramatic changes in the enzyme's ability to act as a therapeutic agent. All right, so how does lysostatin actually work to kill bacteria? So lysostatin, or for that matter, any lytic enzyme, functions in the following way. So here's a false colored electron micrograph of Staph aureus cells. Here's a cartoon schematic of a cutaway of one cell showing a thick outer cell wall. And if you zoomed in on this cell wall, you would see it's composed of a heteropolymer of long glycan chains that are cross-linked by short peptide chains. And so it's this cell wall, this peptidoglycan cell wall, that protects bacteria from the enormous osmotic stress that they experience in the environment. So what I'm going to do is I'm going to show you here a lifetime movie of 2 identical bacterial suspensions. I'm going to add a little bit of lytic enzyme to that right hand to shake it up. And what you're going to see immediately is that that suspension starts to go progressively more clear. And what's happening at a cellular level is that the enzyme is homing in on that bacterial cell, leaving key bonds in the cell wall of glycan, undermining cell integrity and causing those cells to catastrophic life. Right, so beyond this kind of empirical evidence of lysostatin's antibacterial activity, lysostatin has actually undergone extensive preclinical and clinical testing. And it's been proven to be very effective at clearing Staph aureus from human subjects. But this wealth of clinical experience has also demonstrated that lysostatin is in fact immunogenic in animals, including eliciting anti-drug antibodies in human subjects. All right, so the interim conclusion here is that lysostatin is a potent and promising antibacterial candidate, but unfortunately, immunogenicity is a barrier to its clinical translation and widespread deployment. All right, so this now brings us to the engineering design challenge that we set out to tackle, and that is, how do we leverage biotechnology in order to render molecules like lysostatin safe to use as therapeutics? And we do that through a process called deimmunization. All right, so I've already shown you this cellular and molecular recognition pathway that leads to development of anti-drug antibodies. Again, what we want to do is mitigate the risks associated with formation of these anti-drug antibodies and their complexation with our drug. But how we're going to do that is we're actually going to go upstream in this recognition pathway and focus on this molecular recognition event. So we're going to ignore the antibodies for the moment and focus on the fact that what drives the antibody response is molecular recognition of these short peptide fragments, or these patterns that our T cells see in fragments of therapeutic agents. So here we've got a peptide fragment rendered as a red space-filling model bound in the groove of a class 2 MHC protein. And again, immunogenic peptides will be surveilled by cognate T cell receptors. And the idea is that if we can introduce mutations into our original biotherapeutic such that its peptide fragments are no longer bound by class 2 MHC, then we can forestall T cell activation. That means we can prevent B cell maturation, and we can potentially head off formation of these anti-drug antibodies in the first place, leaving our lysostaphin to go into the human body and do its job in clearing those Staph aureus bacteria. And in fact, it turns out that it's a very simple thing to do to deimmunize a peptide, to mutate a peptide so that it's no longer bound by class 2 MHC. The challenge here is that we're not interested in deimmunizing peptides, but rather these immunogenic peptides are in fact integral embedded components of these precision engineered nanomachines that are drugs, such as lysostaphin, such that the mutations we make to reduce the immunogenicity of the peptide fragments have to maintain compatibility with the fully folded, fully functional therapeutic structure. So to kind of show this in a little bit more detail, here's a cartoon schematic. So you can imagine that we've got a wild type protein, maybe lysostaphin. It has a primary structure, so just a series of amino acids that are linked together through peptide bonds. And this primary sequence folds up into this precisely engineered protein structure that has these wonderful therapeutic properties, able to kill drug-resistant Staph aureus. Now, unfortunately, embedded within wild-type lysostaphin are these high-risk patterns that the immune system sees. So these short peptide fragments that constitute T-cell epitopes, those peptides are bound in the groove of class 2 MHC after being processed out of the full-length protein. And it's this molecular recognition by class 2 MHC that elevates the risk of an anti-drug immune response for wild-type lysostaphin. So our job as protein engineers is to identify mutations to these immunogenic subsequences such that they no longer bind class 2 MHC, rendering the protein lower risk for an anti-drug immune response. Now, the difficulty with this is that many of those deimmunizing mutations are going to be incompatible with the properly folded protein structure. So we may have deimmunized the polypeptide, but it no longer folds up into this functional nanomachine. So we have no therapeutic value here. So the challenge again is that we need to find exactly the right mutations to make that both mitigate binding by class 2 MHC and reduce immunogenicity, yet maintain the protein's native fold and its inherent activity and therapeutic value. So led by Chris Bailey-Kellogg, both over at the Dartmouth Computer Science Department and now at our startup company, Stealth Biologics, we have developed integrated protein design and optimization algorithms that leverage computational T-cell epitope predictors to identify what are those high-risk subsequences or the high-risk patterns that the human immune system sees in protein therapeutics. We can use those epitope predictors to also identify what are the mutations we can make to those subsequences in order to hide them from the human immune system. And then the algorithms use statistical sequence potentials and structure-based molecular modeling in order to assess, well, if we make these deimmunizing mutations, what are the structural and functional consequences in the context of the full-length biotherapeutic agent? So this has been a novel contribution to the space. By way of integrating these types of computational tools to develop these fully integrated protein design and optimization algorithms. We combine that with state-of-the-art protein engineering technologies in order to realize those deimmunized therapeutics in the lab. And this is a couple of handfuls of the papers that we've published over the years where we have shown how these algorithms can be leveraged in an experimental setting. All right, so the interim conclusion here is that biotherapeutics can be deimmunized by mutagenically deleting their constituent T-cell epitopes. And I'll emphasize that these dual objective optimization algorithms developed by Chris and his group can accelerate and de-risk the deimmunization process, right? These algorithms balance these two opposing objective functions, wanting to reduce immunogenicity by mutagenesis, but at the same time maintain high-level stability and activity in our biotherapeutics. All right, so now we're going to shift focus to kind of the last part of this talk, and that is a case study of how we have leveraged this type of technology to try to develop a next-gen antibiotic. All right, so central question number one is, can lysostaphin be genetically engineered to delete putative human T-cell epitopes while retaining potent antibacterial activity? And so in order to attempt to do this, we have leveraged both the computational design platform that I just told you about, and then again, some state-of-the-art protein engineering technologies in this case, a directed evolution algorithm. So directed evolution is analogous to the design, build, test engineering cycle that is frequently brought to bear on engineering projects. What's a little bit different about the directed evolution algorithm is that instead of designing, building, and testing 1 or 2 prototypes at a time, and then refining that 1 or 2 prototypes, in the case of lysostaphin, we designed and built and tested on the order of tens of thousands of different prototypes simultaneously each cycle, and we would find the best possible prototype out of a given cycle, and then use that as a template for a second round or third round or fourth round of subsequent engineering. And through this iterative process, we came up with a stealthy version of lysostaphin that has been given the name LYT100. So LYT100 has about a 5.7% mutation rate. So that's 14 mutations in this 250-ish amino acid protein. I'll note that many enzyme engineering projects accomplish their goals by introducing 1 or 2 or 3 mutations. So pouring 14 mutations into this enzyme was an extremely high mutational load. We did that specifically because we were aiming to delete T-cell epitopes that were scattered throughout this protein structure and sequence in order to render it low immunogenicity for a large fraction of the human population. All right, so the first question is, now that we've poured all of these mutations into the enzyme, does it still maintain its function? Can it kill bacteria? And to do that, we use a test called a minimal inhibitory concentration assay. Essentially what we do is we run some experiments where we look for the lowest concentration of drug that will prevent overnight outgrowth of a MRSA clinical isolate. So for minimal inhibitory concentration assays, the MIC value, or the lower the MIC value, the higher the in vitro potency of the drug. All right, and what I'm showing you here are MIC values for LYT100 when tested against vancomycin-resistant Staph aureus, linezolid-resistant Staph aureus, daptomycin-resistant Staph aureus, and methicillin-resistant Staph aureus. So these are Staph aureus strains that are resistant to all of the standard of care therapies for Staph aureus or drug-resistant Staph aureus infections. And I'll note that the MIC values for LYT100 are in the range of 200 to 400 nanograms per mil. To put that in perspective, when these chemotherapeutics are working, right, their MIC values against drug-sensitive strains are typically on the order of about 1 microgram per mil. So relative to standard of care chemotherapies, we're very potent. As another comparison, here I'm showing you MIC values that we've taken from the literature for a competitive molecule called CF301. So this is a clinical stage lytic enzyme for Staph aureus. It's actually in phase 3 trials in the United States right now. So this is an enzyme that is largely native, so it has not been deimmunized, derived from bacteriophage. And because it has not been deimmunized and because of the results of toxicity testing in preclinical studies, CF301 is being developed for once only dosing to human patients. So you can get this drug one time and that's it, because of concerns about the risks of developing an immune response against it. And when you look at the MIC values for CF301 compared to LYT100, we are more than an order of magnitude more potent in vitro. And I'll note that CF301 is incredibly effective against certain Staph aureus infections in the clinic, and yet we are more potent in vitro. All right, so the interim conclusion here is that LYT100 is a highly engineered lysostaphin variant that we've developed for reduced T-cell epitope content. And despite its high mutational load, LYT100 maintains potent in vitro activity against a wide range of drug-resistant Staph aureus clinical isolates. All right, so that's good, right? That's part of our objective, is that we didn't want to disturb the enzyme's inherent therapeutic activity. But I'll note that we didn't put 14 mutations into lysostaphin just to maintain its activity. We put those mutations in because we wanted to deimmunize it. All right, so this leads us to the next question then, and that is, well, does LYT100 evade human immune cell surveillance, right? Can it stop T-cell activation in human blood samples? And so to do that, we use some ex vivo tissue culture techniques where we take what are called peripheral blood mononuclear cells. These are essentially mixed immune cells from healthy human donors. We take those cells and we put them in tissue culture and expose them to, in this case, wild type lysostaphin. So for each subject, there's going to be professional antigen presenting cells that can internalize that lysostaphin, unfold it, chop it down into peptides, present those peptides. And if that donor is able to recognize those high risk patterns represented by these peptide T cell epitopes, then the T cells will be stimulated to expand over the course of a 2-week incubation. And at the end of this expansion of the antigen-specific T cells, we can come in and challenge with peptides derived from the wild-type lysostaphin. And we can then quantify how many activated T cells did this blood cell donor have using a technique called ELISpot. And what's really helpful for us using this technique is that we can simply take another vial of blood cells from that same human donor and run a parallel experiment where we expose those cells not to wild-type lysostaphin, but to this de-immunized variant. And the expectation is that if we successfully de-immunize lysostaphin, then we'll have less expansion of the T cells, and we'll read that out in our ELISpot assay. So here I'm showing you the results of a PBMC assay that we ran. On the y-axis is a stimulation index. This is a measure of the extent to which T cells are stimulated. So higher stimulation index means a stronger T cell response by that blood cell donor. And I'm showing you stimulation index for wild-type lysostaphin in gray and LYT100 in green. And on the x-axis, I'm showing you the MHC II genotype for 12 different blood cell donors that showed any measurable response against either one of these proteins. And what's immediately obvious from this bar graph is that there are a large number of donors that show a strong T cell response against wild-type lysostaphin, but in general, the LYT100 silences those T cell responses. All right, so the interim conclusion is really just that wild-type lysostaphin activates T cells among a genetically diverse panel of human blood cell donors in our experiments. And this is consistent with the clinical experience that lysostaphin elicits antibodies in humans. Now, at the same time, LYT100 generally silences activation of T cells in those same donors. So it now appears, or at least we have evidence, that we have achieved another one of our objectives, right? We sought out to re-engineer lysostaphin to delete those T cell epitopes and silence T cell activation. And this is evidence, preclinical evidence, that we've achieved that. Now, I will note, however, that ultimately, I really don't care about silencing T cell activation, except for the fact that that is a necessary step towards addressing the anti-drug antibody response. So my real concern here, once I get to the clinic, is have I prevented formation of anti-drug antibodies? And that leads us to the next question, which is, well, does LYT100 evade the T cell mediated anti-drug antibody response? And the only way to answer this question, or the most facile way to answer this question, is in a living system. But keep in mind that we've engineered LYT100 specifically to be de-immunized for humans. And this is a preclinical drug candidate. So to answer this question about the antibody response or evasion of the antibody response, we've leveraged genetically engineered mice. So these are mice that have partially humanized immune system. So these class 2 MHC transgenic or HLA transgenic mice have functioning cellular immune systems and humoral immunity. They can develop anti-drug antibodies against a protein upon immunization, but they do so by presenting those high-risk patterns or those T cell epitopes using class 2 MHC that's derived from humans. So they've been genetically engrafted with a human form of class 2 MHC. All right, so here's how we assay whether or not we've mitigated the anti-drug antibody response. We take 2 groups of our humanized HLA transgenic mice and we immunize them once a week for 4 weeks with either wild-type lysostaphin in gray or LYT100 in green. And then we collect blood from these mice at intermediate time points, and we simply check for anti-drug antibodies in those blood samples. So here I'm showing you the ELISA data for the day 14 blood collection. So essentially in this experiment, the higher these data points on the y-axis, the stronger the antibody response in that blood sample. And what I'm showing on the x-axis is simply a serial dilution of the blood sample from each of these mice. So if we maintain high level signal out here farther out to the right on the x-axis, that's indicative of a very strong antibody response against a protein. And so what you see on day 14 here is that mice being immunized with wild-type lysostaphin in gray are starting to develop an anti-drug antibody response, whereas the vast majority of the mice receiving LYT100 are essentially down at baseline levels of anti-drug antibodies. By day 28 of this study, all of the mice receiving wild-type lysostaphin have developed high titer anti-drug antibodies, whereas mice receiving LYT100 are on average more than an order of magnitude lower in their anti-drug antibody response. All right, so the interim conclusion here is that LYT100 silencing of T cell activation yields dramatically reduced anti-drug antibody titers in humanized HLA transgenic mice. So this is a clinically relevant animal model of immunogenicity or T cell driven immunogenicity. And I'll note that I showed you data only for the DR4 transgenic mouse strain. We actually have 2 transgenic mouse strains, and we get similar results in both of those strains. All right, so I've now shown you that we can pour mutations into lysostaphin yet maintain good antibacterial activity in vitro. I've shown you that those mutations in fact silence human T cell activation, and I've shown you that by silencing T cell activation, we have reduced the anti-drug antibody response in humanized mice. But at the end of the day, maybe the most important question, if we are really focused on getting this molecule into the clinic, is to ask, is LYT100 efficacious in vivo? Can we treat life-threatening Staph aureus infections in vivo? And so to answer that question, we've actually run a couple of different models. So one of these models is a preclinical model of left-sided infective endocarditis. So this is a treatment-refractory Staph aureus infection. It's a Staph aureus infection of the heart valve. So the bacteria actually starts to form biofilms on the heart valves. The left-sided infective endocarditis is more difficult to treat than the right-sided. Mortality rates in general for invasive MRSA infections are in the order of 24 to 44%, depending on the location and the context. The average length of hospital stay for these types of invasive infections is 21 days, and the average healthcare costs or excess healthcare costs is far in excess of $100,000 per patient. So these are very serious, life-threatening, and costly infections, and again, very difficult to treat. All right, so this is a preclinical model that is run in rabbits. The way that the model works is that the rabbits are surgically implanted with a catheter in their heart. After surgical recovery, they're given an infection by a MRSA clinical isolate. That MRSA establishes a biofilm infection on the heart and on the catheter in the heart. And then over the course of either 3 to 4 days, depending on the details of this study, those rabbits undergo a treatment regime with various antibiotics. And at the end of the study, we sacrifice the rabbits and we collect various tissues. We collect the heart, the kidney, the spleen, and the blood. And we look for how many viable MRSA bacteria are present in each of those tissues. So here in blue, what I'm showing you is the number of viable MRSA bacteria that are isolated per gram of tissue. And I'm showing you this in blue for cardiac vegetations. Note this is a log scale. So the treatment groups that I'm showing are controls. And so for the controls, right, we're generating about 100 million bacteria per gram of tissue. We also have a daptomycin treatment. Daptomycin is a standard of care therapy for MRSA infections. And so we achieve about a 1.5 to 2 log knockdown of the bacterial burden in the heart with daptomycin. With LYT100 as a monotherapy, again, we get about a 1.5 log knockdown relative to no treatment. But the just super compelling result here is that when we combine daptomycin and LYT100 at these 2 concentrations, we completely eradicate MRSA from the heart valves of those rabbits. You cannot detect viable MRSA in the hearts of those rabbits. We also, as I said, collect kidneys, and the trend is the same. We completely sterilize bacteria in the kidneys of the combination therapy rabbits, completely sterilize bacteria in the spleens of the combination therapy rabbits, and completely sterilize bacteria in the blood of the combination therapy rabbits. We also rescue every one of the rabbits in that combined treatment group. So this shows that LYT100 synergizes in vivo with standard of care therapies, and it actually points directly to a means of conducting clinical trials, and that is we just layer LYT100 right on top of the standard of care therapies that patients would get anyways, and we looked for an improved outcome. So I think this is a super compelling result because this infective endocarditis is one of the most difficult infections to treat. But the perhaps more important outcome that we've gotten as far as in vivo efficacy relates to the capacity to redose LYT100. I told you that there are actually lytic enzymes for MRSA that are in the clinic, but they're approved to be dosed once and only once to each patient because of concerns about immunogenicity. Our goal with LYT100 is to develop an enzyme that can be dosed multiple times to treat the most refractory and most deep-seated MRSA infections. And so to see whether or not we might be able to do that, we have leveraged a disease model, a recurrent disease model in our humanized HLA transgenic mice. So again, these are mice with partially humanized immune systems. This is a recurrent bacteremia model wherein we give the mice a lethal challenge with a MRSA clinical isolate. 1 hour later, we come in with a single systemic bolus treatment of either LYT100 in green or wild-type lysostaphin in gray. And mice that survive this first infection in treatment are given a week to recover, and then they're reinfected and retreated. Mice surviving the second infection are given a week to recover, and they're reinfected and retreated. And we do this in a serial fashion for a month and a half. All right, so here's the result of this recurrent infection study. There's a Kaplan-Meier curve showing the survival for groups that are treated with wild-type lysostaphin in gray. And you see that by day 21 of this study, all of those animals had become moribund and had to be sacrificed, whereas all of the animals treated with LYT100 survived throughout the full course of this month-and-a-half study of repeat dosing. And so the important point here is that if the immune systems of these humanized mice were mounting a response against these drugs, we would expect to see the efficacy fall off. And that is not what we see with LYT100. LYT100 maintains its efficacy throughout the length of this study. We've repeated this recurrent infection study a couple of times, and during a repeat of the study, which gave largely the same results, we collected blood at an intermediate time point, day 21 that time. And so the dramatically improved efficacy of LYT100 compared to wild-type lysostaphin at these later time points correlates with a substantial reduction, a striking reduction in the anti-drug antibody titers against the deimmunized enzyme. All right, so hopefully I have shown you some convincing data that in fact we have generated a deimmunized version of lysostaphin. I'll note that the data that I've shown you today is really just a fraction of the data that we published in this recent Science Advances article. We've done much more extensive immunological analysis of this enzyme as detailed in this paper. I'll note that we have 2 other papers that are currently in press at Antimicrobial Agents and Chemotherapy that detail other studies of this enzyme, exploring and examining the clinical potential of this agent. So this is an ongoing development program. The enabling IP for LYT100 and related lytic enzymes has been licensed by our startup company, Liticon LLC. And the commercial development of that enzyme is ongoing. And I'm hopeful that in the near future, we may be in a position to actually see this molecule go into a first-in-humans trial. All right, so maybe stepping back a little bit, though, let me give you some big picture conclusions here. So one of the first ones is, beyond lysostaphin, just in general, protein therapeutics can elicit detrimental anti-drug immune responses that cause all kinds of complications in the clinic. But protein therapeutics can be deimmunized by mutagenic T-cell epitope deletion. I'll note that lytic enzymes in particular represent a very promising therapeutic avenue for treating multi-drug resistant bacterial infections and addressing the health crisis of multi-drug resistant bacteria. But unfortunately, all of the enzymes that are in development right now are derived from phage, and they are known to be immunogenic in preclinical animal models, and those that are in the clinic have proven highly immunogenic in humans. All right, so my goal here today was to show you that in general, we can leverage our combined computational and experimental platform in order to functionally deplete putative T-cell epitopes from a biotherapeutic agent, right? That is, delete those T-cell epitopes to hide those dangerous patterns from the human immune system, but at the same time, maintain the functionality of those therapeutic candidates. I'll also emphasize that I only care about deleting T-cell epitopes insofar as it allows me to reduce anti-drug antibody responses in vivo. And I've shown you data in humanized mice that in fact, we have achieved that goal. And again, I only care about reducing the anti-drug antibody response insofar that doing so will yield a better biotherapeutic agent. It will yield a safer biotherapeutic and a biotherapeutic that has enhanced therapeutic efficacy. And I'll note that our collaborative effort is, to our knowledge, the very first direct demonstration that deimmunization by T-cell epitope deletion results in improved efficacy in a clinically relevant humanized animal model. All right, so with that, I'm going to take just a couple of minutes to thank the folks that really did the heavy lifting on this project. So in my lab at Thayer, Dr. Hongling Zhao led the protein engineering efforts. Dr. Seth Brooks led the immunogenicity analysis efforts, as well as some of the in vivo efficacy. Dr. Susan Esterhaus has spearheaded the in vivo efficacy work, as well as contributing to the immunological analysis. A former graduate student, Youngling Fong, actually headed up a team of very talented undergraduates that did some great in vitro characterization of this drug candidate. In the computer science department, Chris Bailey Kellogg, with assistance from two great postdocs, Deeptak Verma and Yoonju Choi, as well as foundational work by a former graduate student, Andrew Parker, led the development of the deimmunization algorithms. Likewise, at Stealth, Chris and I have been assisted by a former scientist, Dr. Hao Chang, who's actually a former graduate student of Margie Ackerman's. And then the in vivo rabbit studies were conducted at the Lundquist Institute under the direction of Professor Yan Zhang, with her postdoc at the time, Lang Li. Let me also acknowledge that our humanized mouse colonies are cared for by Steve Firing and Jennifer Fields. The really important work that we do with those powerful models wouldn't be possible without them. Funding for this work is from the NIH predominantly, as well as the NHIRC, and those are grants to both Liticon as well as Stealth Biologics. And with that, I will also thank all of you for joining me this afternoon.